As the global population heads toward 10 billion, the pressure on agriculture is mounting. With that in mind, the UK's Advanced Research and Invention Agency has announced millions of pounds worth of funding for a research programme aimed at developing synthetic plants. These are crops enhanced through synthetic biology by designing entirely new chromosomes and chloroplasts. Starting with the potato, researchers hope to create plants that are more productive, resilient, and sustainable. This ambitious work could transform how we grow food in the face of climate change and resource shortages. The program also includes dedicated research into the ethical, social, and ecological dimensions of these technologies. We'll be speaking with Angie Burnett, an ARIA program director and plant biologist leading this initiative, to find out how synthetic plants could help reimagine the future of farming.
The way that I like to think about this is if you could imagine that the DNA of a plant was actually a book. And if you're thinking about techniques which you can use to change what plants are doing, in traditional breeding, you're essentially swapping little parts of the book around, but in a non-targeted way. So that's just by combining the genomes of plants together when the plant breeds. And you'll end up with a reshuffling. So it's like you have two different books and you're changing some of the information between them and getting out a new book at the end. What gene editing is like doing is it's a bit like if you were going to change one letter in a book, very, very precise. When you think about genetic modification, in that case, it's changing entire paragraphs, moving sections around within the book. Again, it's quite targeted in terms of what is being done. Although, unlike gene editing, it's not always easy to control exactly where those changes are happening. With synthetic plants, so the technology that we're developing in our synthetic plants program, instead, it's like writing a whole new chapter of the book and adding it in like an appendix. And so the great thing about that is that you're able to create potentially something very complex in terms of a functionality, which might rely on lots of different genes, much more complex than the one that can readily be modified using gene editing or genetic modification or breeding.
That is a beautiful analogy. I love that. How is this being done? What exactly is ARIA doing in the lab to make this happen?
ARIA is funding 9 projects within this program. Some of them are looking at doing this for a chromosome. Some of them are looking at doing this for a chloroplast. A lot of this work is computational and really thinking about, okay, what would be the genetic material needed to make a new chromosome or to make a new chloroplast? What might be some of the characteristics that we would want that to have? And in plants, there are some really important characteristics that are really difficult to improve upon using genetic modification or gene editing or breeding. Traits like yield and photosynthesis, which rely on a lot of genes all over the genome. So those are good target traits. And then thinking about possibilities to have switches, biological switches, which could activate the unit or deactivate it. Then when we think about building, there's been amazing advances in DNA synthesis in recent decades. So these units are going to be substantial amounts of DNA. So for the chromosomes, we're looking for up to 1 megabase. So that's quite a big stretch of DNA. And then the third area that we have is delivering. Delivering DNA into plant cells, which is a process called transformation, has historically been very challenging. And I think that what we expect to see is a real step change, a real paradigm shift in what's possible in this area, which will benefit not just the work of this program, but also be a really fantastic thing for the plant biology community worldwide if we can help to kind of overcome some of those bottlenecks.
And this type of technology would be able to help crops become more resistant to pests, climate, that type of thing?
Absolutely, yeah. So this is a really strong motivator for me, something I'm really excited about. Climate change is actually projected to decrease the yield of our major crops by 11%. So that's a really big decrease. While at the same time, we expect that our global population will be almost 10 billion people by 2050, and we're going to need 70% more food in order to be able to feed our population. And so we need to think about a new generation of crops that are sustainable, that are resilient, and that are productive. And when we think about climate change, there's quite a few different stresses that places on plants. For example, we have drought stress. Plants are susceptible to heat stress as well. The distribution of pests and pathogens is changing as well. And then in terms of water use, for example, across the globe, agriculture uses 70% of global fresh water. So if we can think about ways to make crops that are able to be resilient to pest and pathogens, are able to be resilient to drought, maybe they can flourish with a much lower amount of water, then this could be really helpful as our population is growing.
Okay, and if you could briefly just tell me about what concerns do people usually have about synthetic biology in food? How are these being addressed?
We recognise that this is a really important area and it's important to understand what people think. So the way this is being addressed within the programme is engaging with the public, engaging with a range of stakeholders. So this includes farmers, supply chain stakeholders, people involved in governance, to really understand kind of which benefits of these plants could be considered credible and acceptable, and under which circumstances that might be. So we're expecting to see a lot of innovation in this area, which will help us understand what people think and give us the opportunity to put that into practice in terms of how this technology could be used in the future.